Hello everyone, and welcome to today's live broadcast. Lab-on-a-Printer, a multi-material 3D bioprinting platform for building living tissues on demand, presented by Dr. Samuel Wadsworth, Chief Scientific Officer and co-founder of Aspect Biosystems. I am Marjorie Torres of LabRoots, and I'll be your moderator for today's event. We are delighted to bring you this educational web seminar presented by LabRoots. LabRoots is a leading scientific social networking website and producer of educational virtual events and webinars. Before we begin, I would like to remind everyone that this event is interactive. We encourage you to participate by submitting as many questions as you want at any time you want during the presentation. Just click on the green Q&A button located in the lower left of the presentation window and type your question into the box that appears on the screen. We'll answer as many questions as we have time for at the end of the presentation. Also, please notice that you will be viewing the presentation in the slide window. To enlarge the window, just click on the screen icon located in the lower right. If you have trouble seeing or hearing the presentation, please click on the support button at the top right of the presentation window or use the Q&A button to let us know that you're having a problem. This presentation is educational and thus offers continuing education credits. Please click on the button in the bottom left corner and follow the process to obtain your credits. Please join me in welcoming Dr. Samuel Wadsworth. I will now turn the presentation over to him.
Well, thank you very much. Hello, everyone. My name is Sam Wadsworth. I'm co-founder and Chief Scientific Officer of Aspect Biosystems. And thank you for the opportunity to tell you about our Lab-on-a-Printer bioprinting technology today. Before I discuss the details of our unique platform technology, I'll talk about why we want to bioprint in the first place and how we think it will improve the way we treat disease and develop therapeutics. Although we've come a long way since the very first successful organ transplant in 1952, with many lives saved, we're faced with a widening gap between donor organ supply and demand. Currently, approximately 122,000 people are on the organ donor waiting list in the USA, and an estimated 22 people die every day waiting for a donor organ. We're several years away and further scientific and technological advancement will be needed, but bioprinting technology holds the potential to enable the production of human tissues for implantation, allowing us to close the gap between organ supply and demand. Before bioprinting technology is used to tackle the bold challenge of donor organ shortage, there are near-term applications of bioprinting technology that could significantly impact the way we treat disease and develop drugs. If we look at the current drug development process, we see that the pharmaceutical industry relies heavily on the use of simple two-dimensional cell cultures and animal models to investigate novel therapeutics during the preclinical phase of testing. Candidate molecules that show promise are advanced into clinical trials where they're tested in humans. And this is where the problems really start. Over 90% of drugs end up failing in clinical trials, with each late-stage failure costing about $200 million. That means of the estimated $3 to $4 billion it takes to develop one successful drug, about $1.8 billion goes towards drugs that never make it to the market, and importantly, never end up saving or improving lives. With simple two-dimensional cell cultures, we often cannot capture the relevant complexity of the in vivo microenvironment, and animal models often misrepresent the human response for many disease indications. With 3D bioprinted tissues, we can better mimic the signals from the microenvironment observed in native tissues and study cell-to-cell and cell extracellular matrix interactions in a human-relevant system, and ultimately bridge the translational gap between preclinical studies and clinical trials. Our goal is that improving the accuracy of the preclinical testing phase will identify drugs that will fail early before expensive clinical trials. This improvement will get better drugs to patients faster at a lower development cost. So our vision at Aspect builds on very rapid advancements in the areas of cell biology, biomaterial science, bio and tissue engineering. Our highly interdisciplinary team at Aspect has developed one of the world's most advanced biofabrication technologies that allows us to target key applications in drug development, cosmetics testing, personalized medicine, and regenerative medicine. This biofabrication technology is based around 3D bioprinting, and I'll now discuss this in more detail. So what is 3D printing? It's also called additive manufacturing, and this is where a software design for a 3D structure is fabricated one layer at a time to produce that physical object. Substances such as plastics are heated up and melted and then extruded from nozzles to fabricate an inanimate 3D structure in a layer-by-layer manner. This is a picture of a common desktop 3D printer from MakerBot, and you can see that it's produced a plastic model of a house. So what is bioprinting? Well, in some ways it's similar to standard 3D printing in the sense that it also builds a physical structure based on a digital design. But that's really where the similarity ends. Bioprinting is different because instead of using plastics, we print with a bioink that contains living cells and biological scaffolds to fabricate three-dimensional living structures. Now, there are multiple approaches to bioprinting, each with its own advantages and disadvantages. Inkjet was the first bioprinting technology developed, and this was really adapted from existing two-dimensional inkjet technology. This is ideal for printing low-viscosity bioinks. Laser-assisted bioprinting is where a donor ribbon of gold or titanium has bioink suspended underneath it. A laser pulse generates a microbubble at the interface between the ribbon and the bioink, and this ejects a small droplet onto a collection surface. Extrusion bioprinting is probably the most common type of bioprinting technology in use today. This is where a higher viscosity bioink is extruded through a syringe needle. The shear stresses introduced into the system during that extrusion sometimes reduces cell viability and also restricts the speed of printing. Stereolithography is one more type of bioprinting, and this is where light, often ultraviolet light, is used to cross-link liquid bioinks to form a solid structure. At Aspect, we've developed a 3D printing technology that leverages the rapid advancements being made in microfluidics. We call this unique approach to bioprinting Lab-on-a-Printer. On the left of this slide is a schematic of one of our two material printheads. Multiple materials, hydrogels, are fed into the printhead, and each material channel is valved, allowing for flow control. These bioinks could include biomaterials and hydrogels loaded with cells, growth factors, extracellular matrix, and other bioactive compounds. Right before deposition, a cross-linking fluid is introduced co-axially, and this rapidly cross-links the bioink to generate a solid fiber that is patterned in 3D. On the right-hand side here, you can see a cross-section of the fiber with that cross-linking sheath fluid surrounding it. Excess cross-linking solution is removed by printing on a porous substrate and applying a vacuum. The Lab-on-a-Printer is a new paradigm where we're integrating complex microfluidic functions and lab-on-a-chip technology directly within the printhead itself. This enables real-time material processing during printing and on-the-fly control over printed structure composition. In the video on the left, you can see a four-material Lab-on-a-Printer printhead rapidly switching between those materials. On the right, you can see a time-lapse video of our 3D bioprinter depositing a hydrogel fiber about the width of a human hair to produce a two-material, three-dimensional tubular structure. The LOP technology enables precise control over fiber size. On the left is a close-up image of the focusing intersection between the biomaterial and the cross-linker sheath fluid. By changing the pressure ratios of the sheath fluid and the bioink, we can modulate fiber diameter and even do this on the fly within a printed structure, as shown in the image on the right, where fiber diameter is increasing, moving from left to right. Fiber composition is also precisely controlled. Because each material channel is valved, we have control of material flow and can turn material channels on and off, allowing us to rapidly switch between materials. We can create individual material packets within a fiber of a volume of around 2.5 nanoliters. This enables the precise deposition of small volumes of precious cells. At the fiber level, we can also introduce different compositional and structural features. At the top, you can see Janus fibers, where the fiber itself is composed of two different parallel material regions, one red and one blue dye. By controlling the pressure ratio between the material lines, we can vary the proportion of each material within the fiber. On the bottom of the slide, we demonstrate our ability to create core shell fibers, where the core can be a second material or can be empty to produce a hollow printed fiber. Here we demonstrate a few basic capabilities of the system to generate heterogeneous macrostructures. On the far left, you can see a printed alginate tubular structure. The structure is built by switching between hydrogel inputs as the printer switches layers. The structure is reproduced with high fidelity, as the transition between one material and another does not introduce start-stop errors that could accumulate in tall structures with many material transitions. In addition, the build time does not depend on the number of switching events. This approach can be used to recreate the banded smooth muscle pattern seen in the human airway, for example. In the center, we show that with on-the-fly computer coordination of on-chip processes within the printer toolpath, we can create co-axial structures with clean boundaries and demonstrate coordinated intra-layer material switching. This structure can be used to model the various concentric layers of a tissue like a blood vessel or the intestine. On the right-hand side, we demonstrate interweaving one hydrogel within a solid and thin film of a second hydrogel. This capability can be used as the basis of a vascularized skin or tissue patch. These structures reproduce the intended geometry with high fidelity and are mechanically robust post-printing. So we see an initial opportunity in developing bioprinted tissues that could be used at the preclinical stages of drug development. Now I'll briefly go over a few example 3D tissue models for use in preclinical assays. So the lung is an incredibly complex branching network of tubes. Each tube ends in one of 300 million alveoli per lung, where gas exchange occurs. Now, even though we can bioprint with fine control over macro and microstructures, we can't print something as complex as the human lung yet. But we can print some of the components of the lung in a way that recreates lifelike function. So this is a fluorescent image of the bands of smooth muscle cells that wrap around the airway. And it's these muscles that contract if you have an asthma attack. We decided to model this muscle tissue in vitro by printing airway muscle cells into 3D tubular structures that have the ability to contract. And we call this tissue the 3D bioring. So it takes about 30 seconds to print a bioring tissue. And post-printing, these bioprinted structures are living, and so they have to be matured and grown in specialized culture conditions. The bioprinted muscle cells are viable, and over time, those cells form interconnected three-dimensional networks. And crucially, those printed tissues are able to contract in a similar way to the real human airway. And so they demonstrate a biological and disease-relevant function. Our goal was to use this tissue to recreate an asthma attack in a dish. Now, the 3D bioring tackles the lack of contractile models that mimic airway biology. At present, the gold standard lab-based airway contraction model is the precision-cut lung slice tissue. This is where 200 micron thick sections of living lung tissue are taken, and these tissues have a great advantage in that they contain all of the relevant cell types from the biological tissue. However, disadvantages of the PCLS model include limitations of tissue and donor availability, challenging preparation protocols, and a limited lifespan in culture. And all of these really limit the broad adoption of this assay. Aspect's 3D bioring airway is a 3D bioprinted tissue that contains primary airway smooth muscle cells at approximately 25 million cells per mil. These cells are viable in 3D culture, and the tissues are functional. The bioring has a rapid dose-dependent contractile response to histamine and a similar relaxation response to the anti-asthma drug salbutamol. Cells from different donors can be incorporated, matrix components can be added to manipulate cellular microenvironment, and tissue production is rapid, taking less than a minute per tissue. These tissues are produced with very high repeatability. If you're interested in learning more, then you can find out more on our website, where you can access our poster that was presented at the American Thoracic Society last year. So to validate bioring tissue function, we used the dose response curves for histamine and salbutamol to calculate the sensitivity of the tissue to these compounds. This sensitivity is called the EC50, or the effective dose that gives 50% response. And we compared the EC50s of the 3D biorings to published data for the same compounds in human and guinea pig precision-cut lung slices. This comparison table demonstrates that the bioring has a physiologically appropriate contraction response to histamine and a pharmacologically appropriate relaxation to salbutamol, suggesting our bioprinted bioring tissue is able to generate physiologically relevant data similar to the highly complex precision-cut lung slice tissue assay. So confirming cell viability is always important when dealing with bioprinted tissues. We've modified and optimized viability assays specifically for 3D tissues. On the left, you can see some fluorescent images from a calcine AM propidium iodide assay. This is a fluorescent stain where living cells are shown in green and red cells are dead cells. By counting these stain cells, we can quantify the percentage of cell viability. In the healthy control image, you can see that the majority of the cells here are green, and almost 100% of these cells are viable cells. If we take the same tissue and freeze thaw it at -20 for a few hours to trigger cell death, then you can see that many of the cells in this sample are red and therefore dead. In the center, the LDH, or lactate dehydrogenase assay, this is an assay where we can measure the release of lactate dehydrogenase from dead cells into the surrounding liquid media. The more LDH release, the more dead cells are present. This assay can be measured in a plate-based format, giving us higher throughput compared to the calcine AM PI assay. On the right-hand side of the slide, you can see data from an Alamar blue, or resazurin reduction assay. Here, living cells reduce resazurin into a fluorescent form that's released into the surrounding media. High Alamar blue gives is reflective of high cell viability, and a low signal is representative of low cell viability, as evidenced in the freeze-thaw sample. Again, this is suitable for a plate-based assay format. So the lactate dehydrogenase and Alamar blue assays can be multiplexed to measure dead cells and healthy cells simultaneously, giving a relative measure of cell viability in relatively high throughput. So I gave one example of a functional bioprinted tissue assay with the 3D bioring, but the Lab-on-a-Printer technology is applicable to multiple tissue types and applications. These are two examples of alternative tissues. So on the left-hand side, the 3D liver is a bioprinted liver tissue, and we demonstrate that the HepG2 hepatocyte cell line printed into 3D structures are viable and able to grow. We tested hepatocyte function using albumin release into the surrounding media and measured that by ELISA, and we can demonstrate that the printed tissues really maintain function for at least 21 days post-printing, as seen by extended release of albumin. On the right-hand side, we have a 3D cancer model, and this is where we can really probe the interaction between tumor cells and the surrounding stroma, potentially vital interaction in the regulation of tumor immunogenicity and spreading. So we've developed a multicellular tissue design that contains a core of tumor cells, in this case HeLa cells, ovarian cancer cells, surrounded by healthy stromal cells, in this case dermal fibroblasts. And using this system, we're able to investigate physiological three-dimensional tumor stromal interactions in a very controlled manner in vitro. So we've successfully printed a variety of adherent primary human cells, cell lines, and non-adherent immune cells, in this case cord blood-derived mast cells. All of these cells that we've tested demonstrate high cell viability in our systems and, to some extent, biologically appropriate behavior, including cell spreading, network formation, or colony formation. Now, our long-term goal at Aspect is to develop the most complex of tissues for applications in regenerative medicine. And one application that we're focused on is knee meniscus tissue for transplantation. The meniscus is a common site of injury and has poor healing properties. While complete or partial removal of the meniscus relieves acute pain, it leads to osteoarthritis. Now, we've partnered with a leading orthopedics company on a project to develop 3D printed meniscus tissue for implantation that will engraft into the host tissue and provide a permanent knee meniscus replacement. Now, recreating the heterogeneous orientation of fibers and material composition within the biological meniscus is essential for achieving specific mechanical properties such as tensile and compressive strength. In this video, you will see how we're able to precisely customize fiber patterning and composition using Aspect's proprietary software suite. Cue video. So we're distributing our Lab-on-a-Printer technology broadly through the launch of a commercial bioprinting system called the RX1. And we work with our partners and technology users to provide a complete tissue fabrication solution. So in addition to the bioprinter platform itself, we also supply printheads, bioink matrix, and tissue licenses. These licenses are detailed instruction manuals on how to print, culture, and assay specific pre-validated tissues, such as the 3D bioring. A key part of our strategy at Aspect is to work closely with our academic partners to drive forward the development of impactful tissue applications. This includes access to technology and know-how, as well as supporting grant applications. For example, we'll be working with our initial early platform access partners on applications ranging from developing models of uterine muscle contraction and vascular contraction that build off our airway contractile model, to printing three-dimensional neural tissue for studying neurodegenerative diseases. We'd love to learn more about your applications and discuss how we could support them. So to summarize, Aspect's Lab-on-a-Printer technology is a fundamentally different approach to bioprinting. It's a broadly applicable technology that is able to reliably and rapidly fabricate living macrostructures with precise control over microscale tissue architecture, and also allows control of the cellular microenvironment at the molecular level. This technology is now available to research partners, and we encourage you to contact us with any questions or queries. Lastly, I'd like to acknowledge all the people at Aspect. It's only with a truly world-class interdisciplinary team that we're able to execute on achieving our bold vision, and that's exactly what we're building at Aspect. I'd also like to acknowledge all the support we've received from the different funding agencies, and I'd like to thank you for your attention. Thanks very much.
Thank you, Dr. Wadsworth, for that informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the green Q&A button at the lower left of the presentation window, type your question into the box that appears on your screen, and click on the send button. We'll answer as many of your questions as we have time for. Let's get started. Our first question is, what are the advantages of 3D bioprinting versus seeding cells onto scaffolds?
So seeding cells on scaffolds has certain limitations, and they are reproducibility or lack of reproducibility between samples. Doing cell seeding in a manual way, it's challenging to recreate every tissue in the same way. With bioprinting, we're able to reproduce and create a tissue that's been designed in software, and so that tissue is produced in a very consistent and reliable way. It's also produced in an automated way. So, we can produce many tissues in sequence or even in parallel sometimes to increase efficiency of that tissue production.
What are some advantages of the LOP bioprinting technology versus other bioprinting methods?
I discussed some of the various approaches to bioprinting, and each approach really has its own advantages and disadvantages. With the Lab-on-a-Printer approach, what we've been able to do is really leverage microfluidics to use a co-axial flow focusing technology. And so what this allows us to do is generate a cell-loaded hydrogel fiber on chip, but protect those cells from the shear forces that they would otherwise be exposed to in other types of bioprinting technologies. So really we believe that because there is very little shear stress in our system, we're able to produce tissues with cells at a very high viability and also print at high speeds. So we think we have a very rapid tissue fabrication technology as well.
You mentioned customization of bioinks. How does this work?
So the bioinks that we print can be loaded with a variety of different growth factors, matrix factors, really any soluble bioactive compound. And we supply kind of basic bioinks to our users, and that's a broadly applicable bioink that's been validated with a variety of different cell types. But sometimes there's a cell type that needs a very sort of specific microenvironment in order to grow or differentiate in the right way. And so what we're able to do is really customize those bioinks by adding specific components to those inks to really trigger the correct cell growth and differentiation of those cells.
How does a tissue license work?
So a tissue license can be thought of as a detailed instruction manual on how to print a tissue. So that includes information on things like cell density, cell type, preparing cells, and then also printing parameters such as the pressure settings on chip, And then moving through that kind of tissue process, we also supply information on how to culture those tissues in the appropriate manner, and then also how to assay them. So for example, with the 3D bioink tissue, we provide detailed information on how to perform the contraction assays, and even how to quantify the data that comes out of those assays. So it's a sort of an end-to-end tissue instruction manual.
How do you validate the tissues you print are functional?
So tissue validation really depends on the type of tissue that we're dealing with. So for the contractile bioring airway tissue, what we did there was we tested contraction in response to a variety of compounds that were known previously to cause contraction in human airways. So histamine, for example, is well known to cause airway contraction, and so we used that compound to test the contractile response in our biorings, and did that at various concentrations to calculate the sensitivity of those rings, and then really compare that sensitivity to pre-existing data that comes from either other tissue models or even clinical data. And really, as we develop other tissues, then they're validated in very similar ways. So it's really using a panel of compounds with known effects, known clinical effects, and known preclinical effects, and then testing those in our system to confirm that our tissues basically reproduce those known effects for those compounds.
Okay, we have time for one more question. Can you bioprint stem cells using the LOP?
Yeah, we've printed a variety of different cells, including primary human cells. Some of those are kind of tricky to grow and culture. So we've definitely printed some, relatively fragile cell types using this system. And some of our collaborators and our technology users do have stem cell-based projects that they're focused on. So, for example, one of our users is using induced pluripotent stem cells and printing those to derive neuronal tissues for testing and researching neurodegenerative diseases and testing potential therapeutics for those diseases. So yeah, we're certainly working with people who are interested and using our system for printing stem cells.
I would like to once again thank Dr. Samuel Wadsworth for his presentation. I would also like to thank LabRoots for making today's educational webcast possible. Before we go, I want to let everyone know that today's webcast will be available for on-demand viewing through August 24, 2017. You will receive an email from LabRoots letting you know when this webcast will be available for replay. Please share that announcement with your colleagues who may have missed today's live event. That's all for now. Thank you for joining us. We hope to see you again soon. Goodbye.
Thank you.